Hello and welcome to Bio40 Pre-Lecture 18. Today we're going to continue our discussion of the genetic code. Here's the code again. Last time we talked about Cricketall's work, which demonstrated that there are 3 nucleotides per codon, that the code is non-overlapping, and that it is degenerate. This time, we're going to discuss how we know which codon corresponds to which amino acid. To understand how this was done, we need to look again at the central dogma of molecular biology. Information flows from DNA to RNA to proteins. But if the information is present in the DNA, and proteins are what carry out the key enzymatic functions, why is the RNA intermediate there at all? Its importance was first noted during investigations with eukaryotic cells in the 1950s. DNA is sequestered in the nucleus, but studies of protein synthesis showed this was happening in the cytoplasm. So something had to transmit the information from the DNA to the cytoplasm. And researchers determined that RNA was responsible for this. RNA molecules actually play several different roles in the cell, so the term messenger RNA, or mRNA, was given specifically to the RNA that has this function. Moving genetic information from the nucleus to the cytoplasm cannot be the only reason that mRNA exists, though, since prokaryotic cells use it too, but we'll get to its other roles later. Knowledge of mRNA led to the development of a couple of technologies that were key for helping to uncover the genetic code. First, we learned how to create synthetic mRNAs with simple and known nucleotide sequences. We also developed in vitro protein synthesis systems. By adding the known mRNAs to such systems, and determining the sequence of the polypeptides that were made, we could begin to fill out the codon table. The system was first used this way by Marshall Nirenberg and Heinrich Matthey. Say they took an mRNA sequence that was just repeating uracils, and added it to the in vitro synthesis system. The system would translate the mRNA code to a polypeptide that was just repeating phenylalanines. So UUU, or TTT as it would be for DNA, must code for phenylalanine. The 4 codons that are just a single base repeating were simple to determine this way. CCC is proline, AAA is lysine, and GGG is glycine. Other codons were more complicated. Repeating dinucleotides could help us figure out more. A sequence of mRNA with UC repeating is translated to a repeating sequence of serines and leucines. There are only 2 possible codons from the mRNA sequence here, UCU and CUC. One of them must correspond to serine and the other to lysine, but this information by itself cannot tell us which is which. Repeating trinucleotide sequences get even more complicated. You might think that a poly-UUC sequence would only give 1 amino acid chain, corresponding to the UUC codon. But the in vitro system doesn't control where the mRNA reading frame begins. It might get read as a series of UUC codons, but it could also be read as repeating UCU codons, or CUU codons. As a result, 3 different polypeptides are made, repeating phenylalanines, serines, and leucines. But again, we cannot match the 3 codons to the 3 amino acids. Note that we already saw the codon UUU code for phenylalanine, but this tells us that UUC, UCU, or CUU must also be a phenylalanine codon. This is further proof that the genetic code is degenerate. 4 nucleotide repeats get more complicated still. Repeats of the sequence UUAC creates a repeat of leucine, leucine, threonine, tyrosine. Once again, this by itself cannot tell us which of the 4 possible codons correspond to which amino acid, but we can begin to combine information to definitively assign codons. For instance, the codon CUU appears both in the poly-UUC mRNA and the poly-UUAC mRNA. But leucine is the only amino acid that appears in both translated polypeptides. So CUU is a leucine codon. Many codons could be definitively assigned this way, but there are still some ambiguities. To resolve the remaining ambiguities, Nirenberg worked with Philip Leiter. By this point, more was known about translation. Translation works because there's an adapter molecule called tRNA that base pairs with the mRNA codons and carries the appropriate amino acid for that codon. And large molecular machines called ribosomes mediate the pairing of mRNA and the appropriate tRNA. So Nirenberg and Leiter mixed different tRNAs with short RNAs containing just a single codon. These are called trinucleotides. The amino acids attached to the tRNAs were radiolabeled. Then they added ribosomes and passed the mixture through a filter. When the trinucleotide was mixed with the wrong tRNA, the two would not interact, and all the radioactivity would pass through the filter. but the right tRNA and trinucleotide would pair together and associate with ribosomes. The filters would not let the large ribosomes pass through, so all the radioactivity would be in the filter. Here you see the example with the trinucleotide CUC. CUC does not pair with the serine tRNA, so the RNAs and the attached radioactive amino acid pass through the filter. But when you mix CUC trinucleotides with the leucine tRNA, they pair, associate with ribosomes, and don't pass through the filter. This tells us that CUC is a leucine codon. In this way, they were eventually able to assign every codon in the table. There are some cool features you can see about the organization. It's arranged in certain ways to minimize the effects of mutations. In many cases, any change to the third base in the codon is completely silent, while in many other cases, transversions aren't silent, but transitions are. Altering bases at other positions are rarely silent, but are often neutral. Note that most of the hydrophobic amino acids are all in this column. A mutation in the first base of any of these codons will result in one hydrophobic amino acid being replaced with another, so the effect on the protein is likely to be small. The last big topic I want to address today is collinearity. A consecutive series of codons in a gene corresponds to a consecutive series of amino acids in the gene product. Charles Janowski originally demonstrated this by studying E. coli tryptophan auxotrophs. Specifically, he looked at mutations in the TRPA gene, which encodes the protein tryptophan synthetase. He was able to generate a fine structure map of TRPA mutations, similar to what Benzer did with the R2 gene of T4 bacteriophage. Janowski was also able to sequence the amino acids of the mutant tryptophan synthetase proteins, and he indeed saw that the base sequence of the gene was collinear with the amino acid sequence of the protein. This study resulted in many important observations. Mutations at neighboring nucleotides could alter the same amino acid, and a given amino acid could have several different substitutions, so a codon must consist of multiple nucleotides. Conversely, point mutations at any given base only ever affected a single amino acid. Each nucleotide is only part of one codon, and thus the code is non-overlapping. This means that you could actually observe intracodon recombination give wild-type alleles. Imagine glycine is supposed to be the amino acid at a given position, but you have two mutants, one with an arginine at that position, and one with a glutamate there. Even though the same amino acid is affected, the two mutants can recombine to give a wild-type sequence. Say the original glycine codon is GGA. A G to A substitution in the codon's first position gives AGA for arginine, and a G to A mutation in the second position gives GAA for glutamate. If a recombination event happens to occur right between the first and second positions of the codon, you can actually get the wild-type codon back. The pattern of substitutions Janowski saw was consistent with the layout of the codon table we've already discussed. This was important because all the previously discussed experiments were in vitro, so there were questions about whether these rules truly applied to living organisms. Let's look at the glycine codon we were just talking about, GGA. A single base substitution can only cause certain amino acid substitutions. If the first base is altered, the amino acid can be changed to an arginine or cause early termination. Mutations in the second position can lead to valine, alanine, and glutamate substitutions, and altering the third base will always be silent. The missense mutations Janowski observed at that codon that altered protein function were always to arginine or glutamate. Presumably, other charged amino acids, like lysine and aspartate, would also disrupt function, but no single point mutation could give rise to those substitutions. Likewise, when Janowski collected reversions to his mutants, the revertants he saw depended on the initial mutation. He sometimes saw true revertants, but he also saw the arginine mutant revert by substitutions to isoleucine, threonine, or serine, whereas the glutamate revertants, besides true revertants, were only to alanine or valine. A glutamate to isoleucine mutation would be functional, since we know isoleucine is tolerated there. But Janowski never saw such a revertant because no single mutation can turn a GAA glutamate codon to an isoleucine codon. One last note about this. When he saw the AGA arginine revert to serine, he couldn't be sure if the revertant was to AGC or AGT. Indeed, he probably saw some of both revertants. The uncertainty was because DNA sequencing still wasn't possible at this point, even though protein sequencing was. All of the mutations had to be determined by inference. In addition to just substitutions, Janowski also looked at proflavin-induced frame shifts. Like Cricketall, he made frameshift mutants, and then got revertins through mutations at second sites within the gene. He saw that just the amino acids between the altered codons were affected, and the changes were all consistent with what would be expected by a reading frame shift between the mutated codons. That does it for our coverage of the genetic code. Next time, we'll get into the processes that take us from DNA to proteins. First, transcription. How is mRNA copied from DNA?